Hey everybody, welcome back to our last lecture video in this semester, or in this chapter, or sorry, in this book. We're going to be looking at chapter 24, Metabolic Pathways for Lipids and Amino Acids, and we're going to be going over essentially how they get utilized for energy and how our body makes them. So what we're looking at here is fat cells. Now, this is where we store our triacylglycerols or our triglycerides. And you can see that the book says here that we can store unlimited quantities, which means there's no upper limit to how much fat we can accumulate. There is a lot of energy stored in fat, and so we're going to look at how that energy gets freed up here in a minute. And in case you forgot, triglycerides are these bad boys. So in a nutshell, this is essentially what's going to happen. Digestion of fat is going to start with bile salts. So bile salts will break the fat into little pieces, and they're going to be surrounded by those bile salts, which are very non-polar, right? This is going to create little micelles, or little bubbles of fat, essentially, that can be dissolved in water. Pancreatic lipases, these are enzymes that are going to come in and actually start to hydrolyze our esters, start to break down these triglycerides into what we get monoacylglycerols, or a glyceride molecule with one fatty acid on it and some free fatty acids. We're going to get phospholipids and proteins coating these chopped up pieces, forming chylomicrons, which we talked about a little bit in the chapter where we were talking about cholesterol. And this is structures that are able to transport fat in the aqueous environment of the blood. Fat gets transported basically everywhere. Cells of the heart, muscle, and into fat tissues for storage. Lipases are going to hydrolyze these triglycerides and form, or these remaining chunks, and there are some recombining that happens between the monosubstituted glycerols and the fatty acids. But these will ultimately all be broken down into glycerol and fatty acids. And glycerol can then be oxidized to give energy. Same thing with the fatty acids. And we're going to look at how each of those is treated. Acetyl-CoA is essentially the molecule that we get from fat digestion. And we know that acetyl-CoA is important for sugar digestion. It's one of the intermediates. In fact, it gets formed from the breakdown of pyruvate, if you can remember that. So the general formula is triglycerol into monoglycerol and some fatty acids. Let's talk about how these fats then get utilized, or what exactly triggers this. So when blood glucose is depleted, and when glycogen stores are low, so low blood sugar and glycogen, which is our sugar storage, depleted. I don't know if that's how you spell depleted. So when that's all gone, I don't think there's an A in there. Anyways, so when sugar's low, fat gets the process of fat utilization is stimulated. There are hormones. Glucagon is one of them. Epinephrine's another. And epinephrine. These are secreted into the bloodstream. They bind to the receptors on the membrane of fat tissue, fat cells. And what that does is it triggers a hormone-sensitive lipase. So we know lipases are these compounds that are going to hydrolyze the ester bonds. Fat storage is just, a bunch of triglycerides. So when these hormones activate these lipases, they start to catalyze hydrolysis of triglycerides. Um, they produce glycerol and they produce fatty acids. This is the same thing as what happens is when we start eating fats and breaking them down through regular metabolism. So the process of producing glycerol and fatty acids is true whether or not we're breaking down our own fat stores or if we're eating fats in our diet. So the glycerol and the fatty acids then diffuse into the bloodstream and they bind with the plasma proteins to be transported to the tissues, muscles, fat cells, wherever they're destined to go. And these could be chylomicrons, these could be LDLs and HDLs to some extent. They're just carrier proteins. Now, glycerol from the fat digestion, if we take a look at what happens here specifically to the structure, let me go ahead and clear this. Glycerol is going to require some ATP activation, and glycerol becomes glycerol 3-phosphate. In the process, we're also going to generate some NADH. Glycerol 3-phosphate and an NAD plus molecule is going to be formed or turned into NADH rather, and the glycerol 3-phosphate will become dihydroxyacetone phosphate. And you might remember this as an intermediate in glycolysis. This is when we finally took that 6-carbon sugar and broke it into 2 pieces, we end up with dihydroxyacetone phosphate as one of those pieces, and we convert that into, I think it's glyceraldehyde 3-phosphate, and that then goes on through the rest of glycolysis. We also learned when we're making glucoses that we can utilize this compound as well. So dihydroxyacetone phosphate is the ultimate fate here for glycerol. Now, the fatty acid side is going to yield some acetyl-CoAs, and so we're going to take a look at how that works. There's a lot of energy in fat, a lot more energy in fat than there is in sugar. Now, oxidation is going to happen in the mitochondria. This means that we're going to get some rounds of electron transport chain, probably going to get some electron carriers here too, to generate some ATP from the oxidative phosphorylation. Each fatty acid has to go through a series of reactions in order to be completely broken down, because remember, these things often have many carbons. The way that this works is next to our carbonyl carbon, we have an alpha and a beta carbon. So the alpha and the beta carbons are essentially going to be removed through a process called beta oxidation. And we're going to just be cutting off 2 carbons at a time. These are what are going to generate our acetyl-CoAs. And then the cycle, we'll go through one sort of round of this, and then you can just sort of imagine doing it over and over to get rid of every carbon. So let's talk about a cycle of beta oxidation. So we're going to produce acetyl-CoA by shortening the fatty acid. And again, 2 carbons at a time. And we'll talk about odd amino or odd numbered fatty acids here in a second, but this process just repeats until the entire fatty acid is broken down, and then it all enters the citric acid cycle. So you can see here the cycle takes place with 4 different reactions. There's an oxidation step. So the FAD coenzyme is turned into an FADH here. This will go on to electron transport chain. In this step, you can see we're basically going to create a double bond here. So it gets I want to say it gets oxidized. Yeah. So it's oxidized. Then there's a hydration step. Here we're going to add water to create an alcohol. So to the double bond, we're adding an OH and an H. So now it's hydrated. Then there's an oxidation again. In this case, we're going to create a keto group in place of that secondary alcohol. So it's going to become a keto group, and we're going to reduce another of our coenzymes that can go on to the electron transport chain. And then now we have cleavage of what we're calling our, well, I guess we don't really call these anything. This is a, here, I have to come up with the name. Oh, I guess there's a name here. It's a ketoacyl, but there's a different name. Let me see if I can remember. Come on. All right, I don't think I'm going to remember. Oh, it's a keto acid. The name I'm trying to come up with is an alpha keto acid, and for some reason, when I saw the word beta, I was thinking that beta keto acid's also a thing, but it's not. Okay, we'll talk about alpha keto acids in a minute. Scratch that I was trying to say anything about this beta here. Okay, so if we can kind of get back to where we were, now we have that first step, right? These first four reactions have to happen. All that has done is created a molecule now that has another ketone group on it, right? We already had one, now we have another, and it's on the beta. So what's going to happen is we're going to have this coenzyme A come in here. Coenzyme A is going to basically attach right here. Wait, that's not what happens. It's going to attach right here. And what we're going to end up with, we can kind of think of as being these molecules, right? It's the CoA part, the S part, and now this, was that the beta? No. There's a new, I'm trying to illustrate what these two pieces are, because we end up with two things that kind of look the same. And I just want to make it clear here. We have a keto attached to some coenzyme A and another keto attached to some coenzyme A. The difference is these are the pieces that are leaving, and they're leaving 2 carbons at a time. It's not the same, like this guy right here that's attached to the fatty acid is leaving with those 2 carbon groups, and the new coenzyme A is coming in here and sort of taking its place. And every time these new coenzyme A's latch on, you see we have less carbons. Now there's 8, and then after another round there's only 6, and then 5, and then finally at the end we'll end up with 2 identical molecules. And again, we get acetyl-CoA. We get acetyl-CoAs at the end. So I was trying to just illustrate where this attaches, but I guess we don't really go over reaction mechanisms in this class, so it's not really gonna be helpful. But again, my point was that this business here is leaving at the acetyl-CoA, or as acetyl-CoA, and this is coming in here and becoming this guy. Okay. But again, I'm not going to ask you guys about the details of this reaction. I just wanted to make this image a little more clear. Some fats, right, in our diet contain unsaturated fatty acids. So I want to talk about those. I did want to mention this too. And I think I had said earlier that all fatty acids were even, and I might have said that in a different, maybe in a lipid chapter. That's not the case. The ones that we make are often even, okay, even numbered, but that doesn't mean every single fatty acid out there is. And so what you can see here is that there are odd number fatty acids, and they go through all the same steps. But when they get to the end, they don't have 2 acetyl-CoAs. What they actually have is a propanyl-CoA, which has 3 carbons in it, and an acetyl-CoA. There is another kind of thing here. This is talking about how many cycles compared to how many acetyl groups get made. And so if you make 6 acetyl groups, I spelled it wrong already. 6 acetyl groups, that required 5 cycles. So it's just one cycle less than the total number of acetyl groups. And what this is saying is just if you have a lot of carbons, then you have to run this cycle over and over, or more than if you had less carbons. So the number of cycles depends on how big your fatty acid was and what fatty acid it was, stearic acid versus linoleic versus palmitic, and that sort of thing. Unlike glycolysis or citric acid cycle that always has 2 cycles per glucose or something set in stone. Okay, so now, some fats have unsaturated fatty acids in them, meaning they have double bonds already. And usually these are cis double bonds. So one of the things that our body does before it breaks these down is that it converts that cis double bond into a trans double bond. This helps get the shape to be a little more right. And then between, and it's important that that double bond basically is between the alpha and beta carbons. And what that does is it essentially forms a molecule that looks like this, right? So it's already sort of been oxidized. It's ready for the hydration step. And that's the point. So these compounds, they already have a double bond. They just need to be converted into trans. Then they can undergo hydration. So essentially they enter in at reaction 2 rather than starting from the very beginning. But because they don't enter in at reaction 1, they don't produce any FADHs. So unsaturated fatty acids don't have as many ATPs that they can make when you compare them to saturated fatty acids. And that's just because the less steps in the reaction cycle and no production of the electron carrier molecule. If we start to total up how many ATPs that we get from breaking down, let's say, I don't even know which fatty acid that we're using as an example here, but let's just say it's capric acid that has 10 carbons in it. So each cycle requires 2 ATPs to get going. Each one, though, produces an NADH and an FADH and an acetyl-CoA. It's every cycle, right? So the NADH we know goes on to make 2.5 ATP. The FADH2 goes on to make 1.5 ATP. And so therefore each beta oxidation cycle that produces these carriers makes 4 ATPs. So already covers the cost of having to put some in. And then that one acetyl-CoA that was produced each cycle enters the citric acid cycle and makes 10 ATP. So if we take if we consider all of those steps and all of the ATP equivalent molecules that are made for something that has 10 carbons in it, we can see that after paying the price to get it started, we get something like 64 ATPs from a fairly small fatty acid, which is already more than double what we would get from a sugar molecule. Now, granted, the sugar molecule doesn't have as many carbons in it, and that's probably the reason, right? These fatty acids are much more carbon rich, and so there's a lot more potential to get energy out of them. But that being said, they are higher energy storage molecules. Now, there is a problem when you only break down fat for energy. And this is something that happens in situations where you're starving or where you are on a carb-free diet or these ketone diets. You get something called ketone bodies, which can build up and cause something called ketosis. And again, diabetes can cause this. Diets that are low in carbohydrates or high in fat, alcoholism, and just starving in general can bring on ketosis. And this picture is here just showing that there are pH strips that you can use to actually see if your body is in ketosis. Let's talk a little bit about some of these things. So if carbohydrates aren't available, then you need to break down fats, right, to meet your energy needs. The acetyl-CoA molecules that we form from fat metabolism or catabolism they produce a lot of acetyl-CoA, more than can be run through the citric acid cycle. So what happens is it starts to build up, and these can combine into molecules called ketone bodies. We're not going to go over all the structures here, but they essentially react together and they form these ketone bodies. And these ketone bodies are what ends up lowering the pH of your blood. The actual pathway is called ketogenesis, which is just the pathway that forms these ketone bodies. But again, they are due to the fact that there's high levels of acetyl-CoA. So ketosis, again, is building up ketone bodies. These acidic ketone bodies lower the pH. And depending on how much lower than 7.4 it is, you're going to have a variety of effects, obviously leading to death. If your pH of your blood changes too much, you can't transport oxygen anymore. So this isn't anything to sort of mess around with. You can usually smell too on somebody's breath if they are in ketosis or doing these ketone bodies. They have sort of a sweet, sort of acetone breath. But anyways, diabetes. We're talking about diabetes here. So diabetes, there's a couple of reasons why diabetes kind of happens. Either you don't make enough insulin, or it doesn't work right, or you don't have good receptors to register that insulin, or actually I don't have diabetes, so I probably could be a little more knowledgeable on it, but I'm not. But I do know that it has to do with your body's ability to utilize the glucose properly and efficiently. So what's supposed to happen is that as as you eat food and your glucose starts to go into your blood, you should detect that. Your pancreas should start to secrete insulin, which is the trigger to your cells to start taking up that glucose. And if you're missing that, then your cells aren't going to get that trigger to, hey, take up the glucose. So in diabetes, the insulin doesn't function properly. If you don't have enough glucose available in your muscle or your liver or your adipose tissue, then several things are going to start to happen, and breaking down fat is going to be initiated, right? You need to get energy. So the glucose levels are insufficient. Your liver cells can actually synthesize glucose from the gluconeogenesis pathway. And this also elevates the levels of acetyl-CoA. So this isn't good for, again, the ketone bodies. Your fats are broken down into acetyl-CoA. Excess acetyl-CoA undergoes ketogenesis. And so we start to get these ketone bodies accumulating. And then once they accumulate, you can go into ketosis. And persons with, uncontrolled diabetes go into ketosis. Let's talk about fatty acid synthesis. So we talked about breakdown. Let's talk about how we form fatty acids. So when we have plenty of energy and glycogen's full, then any excess carbohydrates and fat and that kind of stuff from your diet get used to make new fats. Because once you've already stored up all the sugar you're going to need, you start to store up the better molecules like the fats. So this happens in the cytosol. 2 carbon acetyl units link together to form, two carbon acetyl units, multiples of them. In this case, you would need 8 of them, are ultimately going to link up to form a 16 carbon fatty acid. So just like a 16 carbon fatty acid would break down into, 8 acetyl units over 7 cycles, right, of beta oxidation, the pathway for lipogenesis, which is the formation of these fatty acids, is not just the reversal of that. We're going to talk about this molecule in a second. This is the carrier protein ACP. So we'll look at this in another slide. Give me a sec. So, separate pathway, different enzymes. It is not a reversal of the beta oxidation. This molecule is going to use NADPH to provide hydrogens for our reduction reactions. There's some reduction reactions that happen. And this happens in the cytosol and not inside the mitochondria where oxidation takes place. So part of the separating of these two things is also so that as we start to make these fatty acids, they're not just instantly broken down and then used for energy in the mitochondria. So we're going to see here that as we start to form these fatty acids, there's actually a membrane blocking them from being able to go into the mitochondria. Okay, so we have carrier proteins, and in this acyl carrier protein is what's going to activate our acyl compounds. And there's actually two of them that are going to be activated by this acyl carrier protein. The carrier protein contains a thiol group. That's the SH here. This is the functional part that's going to basically hook on to our acetyl-CoAs and carry them around, and also our malonyl-CoA. And it also has this pantothenic acid group, which is a vitamin B group, that's attached to a protein. So I think it's most of this stuff here, and then here's our protein. So this is not the whole structure, right? All right. So the first thing that has to happen is we need to prepare some of these carrier molecules, and so they need to be activated carriers. And so we need to synthesize something called a malonyl ACP. So a 3-carbon malonyl ACP is made from acetyl-CoA and bicarbonate. Bicarbonate is a molecule in your blood that kind of buffers your blood. Malonyl-CoA gets formed. And this actually costs some ATP. So this is an energy requiring step, but now we have this malonyl-CoA. All right, so now we have some acetyl-CoAs that are still floating around, and now we have this 3-carbon malonyl-CoA. Now we need to get activated forms of both of these. So malonyl ACP and acetyl ACP are produced when those groups combine with that carrier protein. So carrier protein adds to acetyl-CoA, makes acetyl ACP. Carrier protein adds to malonyl-CoA, which we just made from acetyl-CoA and bicarbonate, into malonyl ACP. So 3-carbon molecule, 2-carbon molecule. Okay, now these are activated. They have the activated protein on them. In order to be activated, they had to have the CoA group on them. Okay, here we go. So reaction 1 in fatty acid synthesis takes place by combining the acetyl ACP and the malonyl ACP. Now you would think adding 2 carbons to 3 carbons would make a 5-carbon molecule, but only a 4-carbon molecule is produced. We lose a CO2 in this process and regenerate our carrier protein. So we have this 4-carbon acetoacyl ACP. So we've combined, we already have now a bigger molecule, right? We had 2 and 3, now we have 4 carbons. Reaction 2. This 3-keto-acyl ACP reductase, which is the name of an enzyme, reduces our 3-keto group using NADPH. So our 3-keto group, let's see, do we have a picture previous? So this guy right here was now reduced. So it's no longer a keto, it's an alcohol. So 3-hydroxyl ACP, 3-hydroxyacyl ACP, and we have an oxidized electron carrier. So, okay, so that's reaction 2. So now we didn't change the number of carbons, but we've reduced it. Reaction 3, dehydration. So we're going to remove a water. 3-hydroxyacyl ACP dehydrates, removes this water. We end up with a trans double bond. The molecule is called trans-2-enoyl ACP. All right, now this double bond, that's going to make it ready to start growing. Reaction 4, reduction. The enoyl ACP reductase, which is the enzyme, and using another NADPH to do a reduction, is going to turn our trans-2-enoyl ACP. So it's just going to add, right, some H's to it, turns it into butyryl ACP. So now we have no double bond, 4 carbons, butyryl ACP. Now, butyryl ACP then is going to react with our malonyl ACP. We haven't seen malonyl for a while, right? But these are being formed at the beginning, right? Those are going to react together to form hexanoyl ACP. So did we lose a CO2 there as well? Yeah. Okay. So malonyl, remember, was 3 carbons. Butyryl is 4 carbons. But instead of forming a 7-carbon molecule, we get hexanoyl, which is 6. But a CO2 floated off into space. Now this process with the adding a malonyl, which is a 3-carbon group, but having a CO2 float off into space, means that you only ever add 2 carbons at a time, repeats and repeats and repeats. Okay, so we can form C16, palmitoyl ACP, which will then be hydrolyzed to remove off the carrier protein to form palmitate, and our carrier protein happens after 7 cycles. And so there's an overall equation that I can show you here. 8 acetyl-CoAs, 14 NADPHs, 7 ATPs can form palmitate and some oxidized form of our electron carrier and some, obviously some spent ATP molecules. And so it's kind of a complicated process. There's a lot of priming and prepping. And then once we get to this hexanoyl ACP, then this malonyl guy just adds over and over and over. So that's how we build these fatty acids up. Now, shorter fatty acids just exit the process a little earlier than the end. They don't go the whole 8 cycles or 7 cycles. So that's how we get 10, 12, 14 carbon fatty acids. And longer ones are produced with special enzymes that will go and add 2 more carbon acetyl units to the end of the fatty acid chain. And so everything from less than 16 to more than 16, but again, evens made by adding 2 carbons at a time. Regulation of fatty acid synthesis. How is this regulated? Well, since this happens in fatty acid synthesis happens in adipose tissue and is stimulated by insulin. So when glucose is high, insulin gets released. If insulin's around, that causes cells to move glucose into the cells, and that should stimulate glycolysis. And the oxidation of pyruvate, that produces acetyl-CoA, and then if we're going to be storing acetyl-CoA, right, there basically has to be a lot of sugar around. If there's no sugar around, then the acetyl-CoA is going to enter the citric acid cycle for digestion, to use for energy. But as long as there's sugar around as well, glucose around, and glycogen, and we don't need to be breaking down these acetyl-CoAs, then they'll start being turned into fatty acids and stored. Okay. So yeah, the highlights are that the 2 carbons are added to the growing fatty acid by using those malonyls. Remember, a CO2 floats off every time we do that. And these acyl groups that are formed, I mentioned a little bit ago, they can't go into the mitochondria so that they end up being utilized by the citric acid cycle. Instead, they stay there in the cytosol. So that's fatty acids. Let's talk about proteins. When you have, or when you don't have, I should say, fatty acids and sugars available, then your body needs to start breaking down your proteins in order to generate energy. And so when we start breaking down proteins, right, we get polypeptides. Polypeptides can be digested, and this is of course if we eat them, not necessarily if they're just in our bodies already. But we can break down peptides into smaller peptides, dipeptides, and ultimately amino acids. And the amino acids are what we can really use for energy. We can turn many amino acids into intermediates for the citric acid cycle. So let's take a look at some of those. But for the most part, it follows the same pattern of digestion. Here rather than waiting into the small intestine to begin digestion or starting in the mouth like with sugars, um, the stomach has activated um, enzymes that will start to actually hydrolyze those peptide bonds. And then in the small intestine, there are different activated enzymes which will continue to break those proteins down. And then eventually we get amino acids which can cross over into the bloodstream. Now, the process of breaking down and making proteins is called protein turnover. So that includes how fast you break them down and how fast you make them. And this is because we keep track, or your body sort of keeps track of the nitrogen content in your body. We don't want to have too much nitrogen. Proteins, which are everything from your muscles to your enzymes to your protein, your fat carriers, HDL, LDL, structural components like hair and fingernails, hemoglobin, which are transport, oxygen transporting molecules, and so on. So a whole lot of what are the other ones? Neurotransmitters. We've got even nucleotides, right? Your DNA. All of these things can contain nitrogen. I'm sorry, I was thinking of proteins. I was thinking of nitrogen right now as I'm listing a million things, but proteins, still have a lot of different functions. And while most amino acids are used for proteins, here's where I guess I was going. Other compounds require nitrogen as well, like nucleotides, amino alcohols, hormones, heme in your blood cells, neurotransmitters. So nitrogen containing, so we need amino acids in order to get nitrogen, right? Amino acids. We need them for the nitrogen so that we can make things that have nitrogen in them. But we also want amino acids so that we can make more proteins. Here's a list of some of those nitrogen-containing compounds that we, again, need amino acids for to provide us with the nitrogens that we need to make these. And actually, there's also some amino acids we just need because we can't make them. All right, so our bodies maintain nitrogen balance. So we break down the same amount of protein that we make, essentially. If we have too much protein in our diet, then we have a positive nitrogen balance, and we have extra nitrogen at the end that we end up converting into urea and excreting out, essentially. We can't store that nitrogen. If we don't have enough nitrogen, then we start to have some problems. Same things that happen when you're starving or when you're fasting, and depending on the situation of fats and sugars, you might start to break those down, or you might just be more of, breaking down more of your muscle for energy than you're actually, making proteins, and that you can imagine why that could be bad. If you weren't making compounds that contain nitrogen, again, going back to this list, it doesn't take very long before you're missing things that your body can't run without. You know what I mean? So that doesn't take very long. Starvation starts to affect these nitrogen-containing compounds pretty quickly. Energy can be extracted from amino acids if there are no other sources, like sugars or fats. If they are the only source of energy for too long, obviously, like I just said, essential body tissues start to break down. Your heart muscle is one of the most important ones, and so just not to understate it, there are a lot of proteins that are vital. But if your heart stops beating or beats poorly, then that obviously affects your ability to bring oxygen to your body and transport nutrients and all of that stuff. But okay, if you need to start breaking down amino acids, there's a couple of ways that we can do it. There's a couple of methods: transamination or oxidative deamination, which is the process of removing or transferring, right, NH2 groups, amino groups. And so let's take a look at the structures here. So when we need to start breaking down amino acids for energy, we can remove the alpha amino group to yield an alpha keto acid. And an alpha keto acid can be an intermediate in the citric acid cycle. So right away we've turned an amino acid into something that we need for just regular energy production. Of course, I don't know how good the citric acid cycle is running when we don't have any pyruvates going into it, but we can also make pyruvate, and I'll show you in a minute, from other amino acids. So literally, if there's only amino acids present, we can turn some of those into pyruvate to enter the citric acid cycle, and we can turn other ones into all of the intermediates of the citric acid cycle, like isocitrate, or citrate, or alpha-ketoglutarate, right, or oxaloacetate, because those have to be present as well. And so basically, we can get the citric acid cycle running off of amino acids. Carbon atoms from the amino acids that are used in the citric acid cycle are also used for synthesis of fatty acids, glucose, and, not importantly, ketone bodies. So because we can turn these amino acids into acetyl-CoAs, for example, we can make fatty acids and ketone bodies, but we can also turn them into pyruvates, into dihydroxyacetone phosphates, and into glucoses if we needed to. Most of the amino acids, though, are converted into urea, and I'll show you how that happens in a couple of reactions. The first reaction is the transamination reaction. So what we've got here is our carboxylic acid group, and so here is our alpha carbon, and so this is an alpha amino, and this alpha amino group can be transferred to an alpha keto acid, and usually it's alpha ketoglutarate. I'll show you where alpha ketoglutarate comes from in a second, but alpha ketoglutarate is around, and it'll take that amino and put it on its alpha, and become an alpha amino acid, because it's got the acid group right here, and an alpha amino, right, once you put the NH3 on there. So then notice what happens. The amino acid, right, in this case it was alanine, here's the R group, right, it got turned into pyruvate. So we've already got something to enter the citric acid cycle and give us energy. Alpha ketoglutarate turns into glutamate. So glutamate we know is a neurotransmitter. Glutamate we know is useful for other things. We're going to see here one of the other really good uses of glutamate. So here we have glutamate. In oxidative deamination, we can take glutamate and we can use NAD+ to actually reduce glutamate into alpha ketoglutarate, regenerating the molecule needed, right, for the transamination reaction to turn amino acids into pyruvate. Alpha ketoglutarate is also a citric acid cycle intermediate. But what we've also done is we've generated an electron carrier that now can go and create some more ATP, right? And we've basically reestablished some alpha ketoglutarate. So this reaction can continue to use up amino acids in the transamination. So what are we going to do with all of this this ammonium, right? Because that's how we remove the NH3 from the glutamate, is we remove it as ammonium. Well, ammonium can build up in the body and be no good. It can be toxic. So what our body does is it uses something called the urea cycle, which we're not really looking at all of the reactions here, but it takes it takes ammonium and carbon dioxide, and it creates urea. And urea, you can see here, is it's like a diamide almost. This gets converted. This is the main component of urine, I guess. So with water and other ions and stuff, this urea gets excreted out through the kidneys. So that is where most of the amino acids sort of go. They get transaminased, so that they're just carbon compounds without an amino group. Then they get turned into, or then they get oxidatively deaminased, which again turns them back into an intermediate, frees up some ammonium, and then the ammonium gets excreted out. Excuse me. We can get carbon atoms from amino acids, so we actually can classify them based on the number of carbon atoms. So there are 3 carbon compounds that we can get from amino acids, there are 4 carbon compounds, and there are 5 carbon compounds the three carbon compounds get converted into pyruvate, the 4 carbon compounds get converted into oxaloacetate, and the 5 carbon compounds give us a alpha ketoglutarate. And then in this picture, you'll see there are some amino acids listed in more than one place because they can actually enter these cycles in different places. So we've got a bunch of amino acids, for example, that can be directly converted into pyruvate. Those can either be used for energy or used to generate sugar. We've got others that are more fat related, I would say. These amino acids can be converted into acetyl-CoA. Acetyl-CoA can enter the citric acid cycle, but it can also build up into ketone bodies. And then we have all the amino acids that just become intermediates, whether it's alpha ketoglutarate, succinyl-CoA, fumarate, or oxaloacetate, which again are necessities to keep the citric acid cycle running. So yeah, that's what we can do with most of these amino acids. Now there are some that we need to get from our diet. Let's talk about those in a minute. The amino acids, back on this page, if they're in yellow versus if they're in red or orange. If they're orange, we call them glucogenic because they can generate pyruvate, alpha ketoglutarate, succinyl-CoA, fumarate, oxaloacetate, something that can make glucose. They're called ketogenic if they can be converted into acetyl-CoA, acetoacetyl-CoA, or ultimately ketone bodies. So ketogenic amino acids give us ketone bodies or fatty acids, and glucogenic amino acids will give us glucose if we were using them to run off of. Now, plants and bacteria make all of their amino acids simply from ammonium and nitrate. Now, we can't do that. We make a lot of our amino acids from other amino acids or from sugar compounds or carbon compounds. Humans can only make 11 out of the 20. So these we don't call essential because we can make them. The essential ones are the ones that we cannot make. And so we have to get these from our diets. The non-essential amino acids we do have pathways for. The alpha keto acid carbon skeleton can often be converted into amino acids, the same way that we convert amino acids into molecules that can make sugars. We can take sugars and modify them into compounds that have aminos on them. So usually transamination is the way that we turn non-amino acid molecules into amino acids. So you can see here there are some glycolysis intermediates. 3-phosphoglycerate and pyruvate can both be transaminased into amino acids, and then like serine can be modified to make 2 more amino acids. So you can see kind of how we get all of these. The alpha ketoglutarate and oxaloacetate can give us the aspartates and the glutamates, which ultimately can give us some more amino acids. They're not showing us here where, so phenylalanine is an essential amino acid, but from phenylalanine we can generate tyrosine, which is a non-essential amino acid. So I don't know if you should call tyrosine non-essential if we need an essential ingredient to make it. But anyways, so metabolism, catabolism, and anabolism. 2 words involved in metabolism. There's the breaking down, catabolism, catabolic pathway, breaking down for energy. Anabolism, anabolic pathway, building up or using energy. And I guess they mix and match, right? There are some things, like it says here, where in the bigger picture, when we're building molecules, sometimes things do get broken down in order to build something bigger. So it's not really a strict, anytime we're breaking down, that's catabolism, anytime we're building, it's anabolism, but there is a little bit of overlap. But in general, catabolic pathways form energy, anabolic pathways use energy. And I think that will just about wrap up this chapter. So here's an image here sort of showing you all the different pathways here, the direction that electrons are ultimately trying to go. Down here, catabolic pathways, breaking down, so from proteins, from sugars, from lipids. Lipids, of course, having the two parts, the glycerol and the fatty acid part, once we break them apart. And then anabolic pathways, don't worry so much about knowing what amino acid can be used to make, or what molecule can make one amino acid, and that sort of thing. But just as an overview, you know, see how it's all connected and how we can break down these molecules and we can put them back together. And even if we don't have the right starting materials, we can wiggle things around and kind of turn molecules we do have into molecules we need. This is obviously not a very in-depth view on this stuff. There's a lot more processes that go on, but I hope it was informative. I hope you guys enjoyed it, and I hope it wasn't too difficult. But that's about it. So again, hit me up with the emails if you have any questions, or on Canvas. Otherwise, I will see you guys in probably our last review meeting. But good luck, everybody. Until next time.